Welcome back and welcome to microbiology. In this video, I am going to discuss the topic microbial growth kinetics. This topic is very important for the competitive examination as well as for your semester exam preparation because you can expect at least one numerical problem from this topic. It is very important and vast topic, so I have divided this topic into parts. And this one is the first part. Here I will discuss what is generation time and specific growth rate constant. So let's start. Growth for high organism is called the ordered increased in size, but due to the microscopic nature of the microbes, it's not possible to measure the increment of weight or length of an individual microbes. So the term growth for microbes is called increase in number of cells in a population. They generally grow and reproduce at regular interval when the environment is friendly, when nutrition are abundant and other factors are favorable for their growth. A mother cell divide into 2 daughter cells and each daughter cells again divide into 4 daughter cells. So they grow in an exponential manner. The time required by a particular microbes to double is called generation time. It is very important parameter to measure the growth kinetics of a particular microbes. In order to solve numerical problem from this topic, you have to remember these 4 equations. The first one is very simple. If you know the initial population and the final population in a bacterial culture, you can easily calculate the number of generation completed by the bacteria. Here I have solved one problem. In this problem, the value of N0 and the number of generation, that is N, is given and you have to calculate the value of NT. I have used the equation that I have described in the earlier. You can also calculate the number of generation completed by a bacterial culture with this equation. And the third equation for the calculation of mean growth rate constant, that is called K, it is the number of generation per unit time, that is expressed per hour, and it can be expressed as N by T. And finally, I am going to discuss about the generation time or G. It is generally calculated from this equation or from the semi-log plot of the growth data in this way. Here in this plot, the bacteria takes 2 hours to double, so the value of G is 2 hours. In this plot, a bacterial population required 6 hours to double, or from 2 into 10 to the power 7 to 4 into 10 to the power 7. So the value of G is T by N, that is 6 by 1 or 6. Now it's time to solve some numerical problems that appeared in the previous year GATE or JAM examination. The number of microbes present in the solution is indirectly measured spectrophotometrically. From OD versus time plot, you have to calculate the value of K for second exponential phase in hour. To solve this type of problem, just take any OD value. For example, I have selected OD 0.8 at 11 hours and at 17 hours it becomes double, that is 1.6. So the bacteria require 6 hours to double. So K is equal to N by T or 0.16 per hour. Here in this problem, the value of N0 and NT are given. Time is 5 hours and you have to calculate the number of generations per hour. I have used previously described equation to solve this. Here the value of N is 20, that completed in 10 hours. So in 1 hour, the bacteria completed the number of generation is 2. Here is another solved problem. In this problem, N0, NT and T are given. You have to calculate the doubling time, that is the generation time or G. So I have used this equation to solve this. The value of G is 0.25 hours or 15 minutes. Now you can try to solve some other problems from the previous year GATE examination. If you like this video and you want to watch the next part of this topic, don't forget to subscribe my channel. You can like, you can share and you can comment. And thanks for watching.